Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, so we've now discussed two of the effectors of calcium signaling. Okay, two of the things that are actually going to change when you get calcium signals occurring in the cytoplasm. So we discussed calcium channel independent kinase 2, and we discussed the conventional protein kinase Cs, both of which are going to be activated when you get calcium signals, and both of which are serine threonine kinases, which can go on to phosphorylate the serine and threonine residues in a whole host of downstream targets. Okay, right. What I now want to discuss is the effect of calcium signals on mitochondria. Okay, so the effect of calcium release from the endoplasmic reticulum on mitochondria. So the first thing that I want to talk about before we discuss how when you get calcium signals occurring in the cytoplasm, you also get calcium signals occurring in the mitochondria, is just a basic overview of the mitochondria. So I want to give a basic overview of the mitochondria, then I want to discuss how you generate calcium signals in the mitochondria, okay, and then I want to talk about what the actual effect of the calcium going into the mitochondria is actually going to be, and it's going to overall increase the rate of respiration, basically. Okay, right. So let me start off by discussing the basic structural features of a mitochondria. Okay, so we're going to draw out a mitochondrion. Right, okay, so a mitochondrion then. So mitochondria then, they have an outer membrane and an inner membrane, okay? So much like the nucleus of cells has an outer membrane and an inner membrane, mitochondria also have an outer membrane and an inner membrane. Okay, so I'll draw the outer mitochondrial membrane first. Okay, so this is representing our outer mitochondrial membrane, which I'll be abbreviating down to OMM, but I'll write it out in full at least once. So this is outer, then mitochondrial is the first M, and then the second M is for membrane. Okay, so I'll just colour in the outer mitochondrial membrane in blue. Okay, so we'll have this coloured in blue. Right, then inside of the outer mitochondrial membrane, you're then going to have another membrane, which will be the inner mitochondrial membrane. Okay, now the outer mitochondrial membrane is a nice sort of smooth oval sort of shape, like so. The inner mitochondrial membrane is much less smooth. Okay, it has invaginations inwards. Okay, so here is one of these invaginations of the inner mitochondrial membrane inwards. Here's another one. Okay, we'll have another one here, like so. And these invaginations of the inner mitochondrial membrane inwards, these are known as cristae. Okay, so this is a Christus, the plural is cristae, so I'll have to have it going to multiple. Okay, right. And the purpose of these cristae is just simply to increase the surface area of the inner mitochondrial membrane, because the inner mitochondrial membrane is the site of the electron transport chain. Okay, and the electron transport chain, which we'll discuss further in a moment, is really, really important in generating ATP. Okay, so you want as much electron transport chain as possible. Okay, and therefore you need as much surface area of the inner mitochondrial membrane for this electron transport chain to occur on. So this here, this is the inner mitochondrial membrane, which will abbreviate down to the IMM. So I for inner, okay, and then of course MM once again stands for mitochondrial, and then membrane. Okay, right, and we'll colour in the inner mitochondrial membrane in green. Okay, so this is the inner mitochondrial membrane. Okay, right. Then in between the outer mitochondrial membrane and the inner mitochondrial membrane, you have that space, okay, and this space is also going to be very important in the function of the mitochondria. Okay, this is called the intermembrane space. Okay, so this space here, this is the intermembrane space of our mitochondria. Okay, and this is the space into which protons are going to get pumped. Okay, so basically in the electron transport chain, which we'll see in a moment, there are complexes, multi-protein complexes, which pump protons into the intermembrane space, okay, from this inner cavity of the mitochondrion. And this inner cavity of the mitochondrion is called the mitochondrial matrix. Okay, so this space inside the inner mitochondrial membrane, that's the mitochondrial matrix. And then the space in between the inner mitochondrial membrane and the outer mitochondrial membrane, that's the intermembrane space. And basically, the idea of the electron transport chain is that we use molecules that we have generated through the breakdown of glucose, and we'll come on to that later on, okay, to pump protons into the intermembrane space out of the matrix. So you end up with a very high proton concentration in the intermembrane space and a low proton concentration in the matrix. You also end up with a powerful electrical potential difference across the inner mitochondrial membrane, okay, because if you're moving protons from the matrix into the intermembrane space, you are moving positive charge basically from the matrix into the intermembrane space, and that's going to make the electrical potential of the intermembrane space go up, because wherever you dump positive charge into somewhere, you increase the electrical potential there. So the electrical potential in the intermembrane space is going to go up. Meanwhile, the electrical potential in the matrix is going to go down because we've removed positive charge from the matrix, okay, and therefore we have removed electrical potential basically, we've made that more negative. So now we're going to end up with an electrical potential difference across the inner mitochondrial membrane of around negative 160 millivolts. So normally across the inner mitochondrial membrane you have this electrical potential difference of negative 160 millivolts, which means that the matrix of the mitochondria, the electrical potential inside the matrix, is 160 millivolts lower than the electrical potential of the intermembrane space. So if I jump from here to here, my electrical potential will go down, and it will go down by 160 millivolts. Okay, right. So there's a basic overview of some of the important features of mitochondria. Let's now talk about why, when we release calcium from the endoplasmic reticulum by either calcium puffs or calcium waves, why is it going to have an effect on the mitochondria? Okay, so basically, you find that when you look inside cells, you find that the mitochondria are often positioned close by the endoplasmic reticulum. Okay, so let me just show this here. Okay, so once again I'll draw a picture of a cell here. And what I want to do is I want to show the endoplasmic reticulum and show these junctions between the endoplasmic reticulum and the mitochondria. So here I'm now showing my endoplasmic reticulum once again, okay, which is the continuation of the space in between the inner and outer nuclear membrane. Okay, now I'm going to put a mitochondrion on here. So basically mitochondria are usually close by the ER here. Okay, so if I colour this in, this blob here in green, this is representing my mitochondria. Okay, or my mitochondrion rather, since it's singular here. Okay, and here in blue, this is the endoplasmic reticulum. Okay, so you find that the outer mitochondrial membrane of mitochondria is generally opposed to the membrane of the endoplasmic reticulum, and this sort of a junction is known as an ER for endoplasmic reticulum, mitochondrial junction. Okay, and it's across these ER mitochondrial junctions that calcium being released from the endoplasmic reticulum by, for instance, IP3 receptors can actually get into the mitochondria. Okay, and in fact the mitochondria can end up with very large calcium concentrations in their matrix when you get calcium being released from the endoplasmic reticulum. Okay, so let's study these ER mitochondrial calcium junctions in more detail. Okay, so what I'm going to do is I'm going to redraw out my picture of the mitochondria now with the endoplasmic reticulum membrane opposed to it. Okay, so let me colour this in. So here is my outer mitochondrial membrane, which I'll show in blue once again here. Okay, and it's now opposed to the endoplasmic reticulum membrane here. So this, which I'll colour in yellow here, this is the membrane of the endoplasmic reticulum, and this will be the lumen of the endoplasmic reticulum. So this is the lumen of the endoplasmic reticulum. We've got a little portion of cytoplasm in between the ER membrane here and the outer mitochondrial membrane here in blue. Okay, now I'll just add on the inner mitochondrial membrane here. Okay, like so, with these cristae invaginating into the matrix. Okay, right, and I'll colour in the inner mitochondrial membrane in green again. So we now want to see how, when calcium is being released from the endoplasmic reticulum, so let's imagine that a calcium wave has been propagating across this cell, okay? So all of the IP3 receptors of the cell are going to be opening in this propagating way, okay? We now want to see how calcium being released from the endoplasmic reticulum can end up in the matrix of the mitochondria. Okay, so this is rather nice. Basically, you find that IP3 receptors, some of the IP3 receptors in the endoplasmic reticulum can be in complexes with receptors on the outer mitochondrial membrane. So let me show this. So here, this is an IP3 receptor of the endoplasmic reticulum here. So this is an IP3 receptor, which will be opening at some point in our calcium wave, okay? So let's say it's one that happens to have 4 IP3 molecules bound to it, so it's now going to be opening if we're having a calcium wave propagating across the cell. Okay, now some of these IP3 receptors end up in complexes with channels in the outer mitochondrial membrane. Okay, and I'll draw a little protein that's linking the two of them together here. Okay, so let me give you now the name of these channels in the outer mitochondrial membrane, and these channels are going to allow the calcium that's coming out from the ER lumen through the IP3 receptor to go straight into the intermembrane space of the mitochondria. Okay, so this channel has a rather weird name since it's actually going to be a calcium channel, but it's called the Vidocq. Okay, and this Vidocq stands for voltage dependent anion channel. Okay, however, it is actually going to be a calcium channel on the outer mitochondrial membrane. Okay, so this is voltage dependent anion channel, but it's going to be functioning as a calcium channel for our purposes. Okay, right, so voltage dependent anion channel, and we've coloured it there in turquoise. Okay, so basically the principle here is that the voltage dependent anion channel, it's normally open in the outer mitochondrial membrane, okay? It is slightly voltage dependent, but it doesn't have that much voltage dependence. The criteria that have to be met are usually met, basically, for it to be open, okay? So for our purposes, we're just going to assume that that is open, basically. Okay, so when the IP3 receptor then releases calcium from the ER, the calcium will then be able to go straight from the cytoplasm into the intermembrane space through this Vidocq channel, basically. Okay, now the one other thing that I need to now tell you is what is the name of this protein, which I'm now colouring in yellow here, which is known to link IP3 receptors in the ER membrane to Vidocq channels in the outer mitochondrial membrane. Okay, well this is a protein known as GRP75, the glucose-regulated protein of 75 kilodaltons. Okay, so this is GRP75 here in yellow that binds to IP3 receptors and Vidocq channels and is therefore involved in keeping them close together here. Okay, so this stands for glucose, that's the G, and then regulated, that's the R, and then it's protein, that's the P, and then the 75 is just referring to its weight. Okay, so it's of 75 kilodaltons. Okay, so remember 1 dalton is the weight of a proton. Okay, so 75 kilodaltons means this is a big molecule. Okay, 75,000 the weight of 75,000 protons, basically. Okay, so it's a big, big molecule. Okay, right. So that's one of these links then between channels in the ER membrane that are releasing calcium into the cytoplasm, and then channels in the outer mitochondrial membrane which are allowing calcium in the cytoplasm to go into the intermembrane space of the mitochondrion. Okay, right. So basically, when you're getting calcium being released from the endoplasmic reticulum, you're going to be getting calcium coming into the intermembrane space of the mitochondrion. And now what we want to see is how does that calcium that's coming into the intermembrane space of the mitochondrion actually get into the matrix of the mitochondrion. Okay, so basically, in the inner mitochondrial membrane, we now have another calcium channel. Okay, so I'll put this here. And this is what's known as the Mitochondrial Calcium Uniporter. Okay, and for short, the Mitochondrial Calcium Uniporter is abbreviated down to the MCU. Okay, so this stands for mitochondrial, that's the M, and then it's calcium, that's the C, and then it's uniporter. Okay, and basically this is a channel that allows calcium to leave the intermembrane space of the mitochondrion and go into the matrix of the mitochondrion. So I'll colour this mitochondrial calcium uniporter in red here. Okay, now uniporter, that means that really it's only going to allow calcium to go in one way, okay, and that direction is inwards. So how does this channel only allow calcium to go into the matrix of the mitochondrion? Well, it's not something clever on the level of the actual channel that only allows calcium to move through it in one direction. The reason that the mitochondrial calcium uniporter only really allows calcium to ever go into the matrix is because it's only ever open when the intermembrane space calcium concentration is extremely high. Okay, so if the intermembrane space calcium goes up extremely high, then the MCU will open, otherwise it doesn't open, okay? Which means that when it does open, because calcium concentration is very high in the intermembrane space, you're always going to get calcium going in because the calcium is really high in the intermembrane space, basically. So now the concentration gradient favours calcium going in. You've also got to consider the electrical gradient across the inner mitochondrial membrane, which is negative 60 millivolts. Okay, calcium is a divalent cation. It wants to go where the electrical potential is lower, which in this case is the matrix of the mitochondrion. The matrix is lower by 160 millivolts. Okay, so whenever the MCU actually opens, then it will be that the conditions favour the movement of calcium in, and that's why this thing appears to only allow calcium to go into the matrix. It's not that there is some clever one-way system actually in the pore that means that calcium can only flow through in one direction. Okay, it's just that the channel will only be open when the conditions are such that calcium will be wanting to move into the matrix. Okay, right, so that's the channel then that allows the calcium that's building up in the intermembrane space to then move into the matrix of the mitochondrion. Okay, so basically, when we now get calcium being released from the endoplasmic reticulum, maybe because we've got a calcium wave propagating across the cell, we're going to get lots of calcium going into the matrix of our mitochondrion, okay, or rather we should turn this into the plural. It's going to be happening for loads of mitochondria, basically, all over the cell. Okay, and we will see what the point of this calcium going into the matrix of mitochondria is in a moment. Okay, but for now what I want to tell you about is the reversal of the calcium signal. This is how you generate very high concentrations of calcium in the matrix of the mitochondria. But how do you then get rid of it after the calcium signal in the cytoplasm has gone? Okay, so basically, we've talked about how calcium signals in the cytoplasm can be reversed. Okay, so once the calcium wave is over, the IP3 receptor will close, SERCA will return calcium into the ER, the NCX and the NCKX will remove calcium from the from the cytoplasm into the extracellular fluid. Okay, so calcium will go back down in the cytoplasm. When that occurs, the Vidocq channel will then allow the calcium to move from the intermembrane space back out into the cytoplasm. Okay, so the intermembrane space will then empty basically of calcium. That will cause the MCU to close, but now how do we get the calcium out of the matrix of the mitochondria? Okay, well, you have again a secondary active transporter which can be used to extrude the calcium from the matrix of the mitochondria. So I'll just put this here. Okay, so this is again one of these sodium-calcium exchangers that we saw was present on the cytoplasm of the cell. So I'll colour this sodium-calcium exchanger in turquoise here. So in turquoise, this is a sodium-calcium exchanger. So it's going to be doing the exact same thing on the inner mitochondrial membrane as it did on the cell membrane. So if I just draw a little sample of the inner mitochondrial membrane out here, here it is in green. Here's our sodium-calcium exchanger here. The sodium-calcium exchanger is a secondary active transporter that is going to be extruding a molecule of calcium, okay, or rather an ion of calcium, okay, in exchange for free sodium ions coming into the matrix of the mitochondria. Okay, now why do sodium ions want to come into the matrix of the mitochondria? Well, firstly, you have to remember the electrical gradient across the inner mitochondrial membrane. That is driving sodium ions into the matrix of the mitochondria. But in addition, there is also a sodium gradient across the inner mitochondrial membrane. Now, how do we establish a sodium gradient across the inner mitochondrial membrane? Okay, we know that on the plasma membrane, the sodium concentration gradient is established by the sodium-potassium pump. But what establishes the sodium gradient across the inner mitochondrial membrane? Well, there is an exchanger, basically, another secondary active transporter. Okay, on the plasma membrane, the sodium-potassium pump is an example of a primary active transporter which produces that sodium gradient across the cell membrane. But in the case of the inner mitochondrial membrane, the thing which produces the sodium gradient across the membrane is actually a secondary active transporter. Okay, and it's going to pump or move sodium ions from the from the matrix of the mitochondrion into the intermembrane space in exchange for a proton coming back into the matrix. So we know that the electron transport chain establishes a very powerful proton gradient across the inner mitochondrial membrane. You have much higher proton concentration in the intermembrane space here than in the matrix. Okay, so there's a very powerful gradient favoring the movement of protons back into the matrix. Okay, this exchanger here is going to allow a proton to move back into the matrix from the intermembrane space in exchange for a sodium ion from the matrix being moved back into the intermembrane space, basically. Okay, so that's how we establish a sodium gradient across the inner mitochondrial membrane. Okay, because now sodium is going to build up in the intermembrane space and it will go down in the matrix, and this is why we can then allow sodium to come back into the matrix via our sodium-calcium exchanger here. Okay, and we can use that to extrude the calcium. So what then is this exchanger for sodium and hydrogen ions called? Well, this is quite simply called the sodium-hydrogen exchanger. Okay, and for short, it's abbreviated down to the NHE, like so. Okay, so this stands for sodium, that's the N, proton, that's the H, okay, and then the E is for exchanger. Okay, so whoever names this one decided not to go for the X to represent exchanger, but just use E. Okay, right. So that then is how we extrude the calcium from the matrix of the mitochondrion, okay? And of course, once it's gone back into the intermembrane space, it can then just go out through Vidocq into the cytoplasm, and then it will be cleared from the cytoplasm. Okay, so to summarise what we've seen so far then, when you're getting calcium released from the endoplasmic reticulum by IP3 receptors, you're getting calcium coming into the matrix of the mitochondria through this pathway that we've seen here. It firstly goes into the intermembrane space from the cytoplasm through these Vidocq channels, which are nicely complexed with IP3 receptors, so that the calcium that's being released by IP3 receptors can just go straight through Vidocq into the intermembrane space here, and then calcium building up in the intermembrane space activates the mitochondrial calcium uniporter, the MCU, to open, okay, and then calcium can move through the MCU into the matrix of the mitochondria. Okay, after the calcium signal in the cytoplasm has gone, then what's going to happen is calcium will then move out of the intermembrane space into the cytoplasm through the Vidocq. And then to get it out of the matrix, you now have to use a different mechanism to the MCU, because the MCU will start to close now that intermembrane space calcium is falling. Okay, so you use these sodium-calcium exchangers here, okay, which extrude the calcium from the matrix into the intermembrane space by bringing in free sodiums into the matrix of the mitochondria down their electrochemical gradient. Now, what establishes the concentration gradient of sodium across the inner mitochondrial membrane? Well, we've discussed that is these sodium proton exchangers here, okay, which use the proton gradient, which we know is across the inner mitochondrial membrane, to extrude a sodium ion into the intermembrane space, basically. Okay, right. So what we now want to discuss then is, what does this calcium coming into the matrix of the mitochondria actually do, basically? What's the point of generating these calcium signals within the matrix of mitochondria? Okay, well, the key thing that's going to happen is the calcium is going to increase the rate of respiration, basically. Okay, now why? Well, a lot of the changes that calcium signals produce within cells are changes that require more energy, basically. So often, when you get calcium signals occurring in the cytoplasm of cells, the change in behavior that induces in the cell is one which requires more energy from the cell, basically. Okay, so the cell is going to require more ATP, basically, to do this new behavior that the calcium signal has induced. Okay, so therefore we need to provide this additional energy, and that's why we need to activate the mitochondrial pathways when we get calcium signals occurring in the cytoplasm. Okay, so what I now want to talk about then is which portions of the respiratory cascade are actually activated then by calcium signals. Okay, so what I'm going to do is give you a brief overview then of respiration. Now, of course, we're not going to go through every single step of the respiratory reactions. Okay, it would take hours to do that. Okay, but I'm going to give you a brief overview of what happens in respiration and the involvement of the mitochondria, and then we'll see which bits broadly calcium is going to affect. Okay, so let me draw another picture of my mitochondrion out here then, and we'll now discuss a brief overview of the respiratory reaction. Okay, right, so here is our inner mitochondrial membrane once again here. Okay, so I'll just colour this in. Actually, no, I think I'll leave it the way it is for now. Okay, right, so we will look at the respiration of glucose then. Okay, so the catabolism of glucose. Okay, so you start off with glucose then, and glucose through a whole series of reactions is turned into pyruvate in the cytoplasm of the cell. So this is the portion of respiration that occurs prior to the mitochondrion. Okay, so this conversion of glucose to pyruvate, this is called glycolysis. Okay, so let's just colour this in. So we'll represent the glycolysis pathway as this arrow here in orange. So the first part of respiration is that glucose is turned into pyruvate molecules. Okay, and we've cut out a few steps here, but this is the basic gist of what happens. Glucose is turned into these pyruvate molecules. Okay, then what's going to happen is these pyruvate molecules are going to go to the mitochondria. Okay, they're going to be able to get across the outer mitochondrial membrane easily, okay, because the outer mitochondrial membrane is quite permeable, okay, so it's not difficult to get across the outer mitochondrial membrane. It's much more difficult to get across the inner mitochondrial membrane. That's a much tighter membrane, much less permeable. Okay, so for the pyruvate to now get across the inner mitochondrial membrane, it has to use a massive great protein complex called the pyruvate dehydrogenase complex. Okay, so this is the PDH complex represented here. Okay, and this stands for pyruvate dehydrogenase. So the P is for pyruvate, okay, and the DH is for dehydrogenase. Okay, now pyruvate dehydrogenase complex, then, it's not just one protein, it is a massive great complex of loads of proteins all stuck together, okay, and it's going to move the pyruvate into the matrix of the mitochondria, okay. However, when it does so, it will actually catalyze a reaction with that pyruvate, okay, so when it moves pyruvate into the matrix of the mitochondria, what will actually happen is the pyruvate will be converted into a molecule of acetyl-CoA. Okay, so when the pyruvate arrives in the matrix of the mitochondria, it will no longer actually be pyruvate, it will instead now be acetyl coenzyme A here. Right, and that reaction, that movement of pyruvate into the matrix of the mitochondrion and its conversion into acetyl-CoA, that is called the link reaction of the respiration reaction. Okay, so this is the link reaction here. Okay, so we've had now glycolysis followed by the link reaction. We've now got acetyl-CoA within the matrix of the mitochondria. Okay, now what happens is acetyl-CoA goes through the Krebs cycle, which occurs within the matrix of the mitochondria. Okay, now because I haven't really got space within the matrix of the mitochondria, I'm just going to put this down here, but in reality it's occurring in the matrix of the mitochondria. Okay, so you're now going through the Krebs cycle, and the Krebs cycle produces two key molecules here. It produces reduced NAD molecules, okay, which will denote NADH, and it also produces reduced FAD molecules, which will denote FADH2. Okay, so this is the Krebs cycle here, or the tricarboxylic acid cycle, whatever you want to call it. Okay, and that's a chemical reaction that is occurring within the matrix of the mitochondria. Okay, so within the matrix of the mitochondria, we are now getting reduced NAD produced then and reduced FAD being produced. Okay, now what's going to happen is these reduced NAD and reduced FAD molecules are going to be used by the electron transport chain. Okay, so now what I'm going to do is I'm going to give a basic overview of the electron transport chain. So I'm going to take out a little piece of membrane here, and we're going to have a look at the different complexes involved in the electron transport chain. So I'm bringing out that little piece of membrane here, that little portion of inner mitochondrial membrane here. Okay, right. Now, there are 5 main multi-protein complexes that are involved in using the reduced NAD and the reduced FAD molecules to actually produce ATP. Okay? Now, 4 of them are the main complexes, and then the 5th one is also called ATP synthase. Okay, so 4 of these are involved in passing electrons along, okay, and using energy that's released by the electrons to pump protons into the intermembrane space. Okay, so here I'm drawing the 4 complexes in the mitochondrial membrane. Now, this side of the inner mitochondrial membrane here is the matrix, and this side therefore is the intermembrane space. Okay, so let's go through these 4 complexes here then. So here in orange, this is complex number 1, okay, and again I'll stress the fact that these are not individual proteins, they are massive, great multi-protein complexes. Okay, so we are getting reduced NAD and reduced FAD molecules being produced within the mitochondrial matrix by the Krebs cycle. Okay, reduced NAD and reduced FAD both carry 2 electrons. Reduced NAD also has a proton attached to it. Again, reduced FAD has 2 protons attached to it, but the protons we don't care about. Okay, they're just going to go into the matrix of the mitochondrion once the NAD gives up its electrons to the complexes, or once the FAD gives up its electrons to the complexes. Okay, so basically NAD, reduced NAD molecules, gives up its 2 electrons to complex number 1. Okay, reduced FAD doesn't give up its electrons to complex 1, it's going to give them up to complex 2, and we'll come to that in a moment. Okay, right, so the reduced FAD molecules that you've produced by the Krebs cycle in the mitochondrial matrix are now going to give up their electrons to complex 1 now. And what complex 1 is going to do is it's going to use energy from those 2 electrons to pump 4 protons from the mitochondrial matrix here into the intermembrane space of the mitochondrion. Okay, so it will pump 4 protons from the matrix into the intermembrane space. Then it will pass the 2 electrons on to complex 3 over here. Okay, and we'll come back to what happens in complex 3 in a moment. What I want to move on to now is talking about where reduced FAD is going to give its electrons to, which is to complex 2. Okay, so reduced FAD, unlike reduced NAD, gives its electrons to complex number 2 here. Okay, NAD doesn't involve complex 2 at all. Okay, so reduced FAD is now going to give its 2 electrons to complex 2 here. Okay, now complex 2 doesn't actually pump any protons on its own, basically. It just passes the 2 electrons on to complex 3, which will then pass them on to complex 4, and complex 3 and complex 4 are going to pump electrons. Okay, so this is how these reduced electron carriers, NAD and FAD, are actually going to give up their electrons to the electron transport chain then. Reduced NAD will pass its electrons to complex 1 here, which will use some energy from those 2 electrons to pump 4 protons from the mitochondrial matrix into the intermembrane space, whilst reduced FAD here will give its 2 electrons to complex 2 here, which won't actually pump any protons on its own. And now both complex 1 and complex 2 will pass the 2 electrons that they've received onto complex 3 here. Okay, complex 3 will then pump protons through. Whether it received the electrons from complex 1 or complex 2, it will use the electrons that it's receiving, the 2 electrons that it's receiving, to pump 4 protons through. Okay, so whether the 2 protons came from a complex 1 or whether they came from a complex 2, it doesn't matter. Complex 3 will use the energy of those electrons to pump 4 protons from the matrix to the intermembrane space. Okay, then what will happen is complex 3 will pass the 2 electrons onto complex 4, the final one in this cascade here, which I'll have in green here. Okay, so this is complex 4, and complex 4 will use the energy from these 2 electrons to pump 2 protons into the intermembrane space from the matrix, and then it will give these 2 electrons to a molecule of oxygen, basically. Okay, so if I bring an oxygen molecule up here, what you're basically going to do is you're going to reduce oxygen molecules. You're going to give electrons to oxygen molecules, and this is going to help you create water molecules from the respiration reaction. Okay, so basically what you're going to do is, in essence, you're going to break this double bond between the two oxygens in this dioxygen molecule here. Okay, in those two bonds, in total there were 4 electrons, 2 from this oxygen, 2 from this oxygen. Imagine sending the 2 electrons from this oxygen back to this oxygen, and the 2 electrons from this oxygen back to this oxygen. Okay, and then what you're going to do is to each one of these oxygens, you're going to give 2 electrons from the electron transport chain here, okay, one after the other. So from one electron transport chain, you're giving away 2 electrons, it'll go to one of them. Okay, the other one will now have to wait for another electron transport chain to occur, 2 more electrons will arrive, and they'll go to the other one. Okay, so what you'll overall generate here from 4 electrons coming through this process, so 2 in turn electron transport chains effectively, you will generate 2 O2 minuses. Okay, and then each of these can combine with 2 protons, okay, from the matrix of the mitochondrion. For instance, the protons that we released from these electron carriers, or else other protons just freely diffusing around in the matrix of the mitochondrion. Okay, you will combine these O2 negatives with protons to create water molecules basically, and you'll then create 2 water molecules from each oxygen molecule. Okay, so that's where complex 4 gets rid of these electrons to eventually it reduces oxygen. Okay, right, so to summarise once again then, reduced NAD will go through the 1-3-4 cascade, and its 2 electrons will eventually be given to an oxygen atom. The reduced FAD electrons will go through the 2-3-4 cascade, and its 2 electrons will be given to an oxygen atom to reduce that oxygen atom to an O2 minus oxygen atom. Okay, right. Now you'll notice that the 1-3-4 cascade is going to pump more protons in total from the matrix into the intermembrane space than will the 2-3-4 cascade. Okay, so NAD, reduced NAD is capable of pumping more protons from the matrix to the intermembrane space than reduced FAD. So you'll get more energy from a reduced NAD molecule than from a reduced FAD molecule. Okay, so that then builds up this proton gradient across the inner mitochondrial membrane. And then what you've got is one final complex, which I'll show on this picture here, okay, which is sometimes called complex 5, but probably is more often known as ATP synthase. Okay, and this is the opposite one to all the others. Okay, all the others were about moving protons from the matrix to the intermembrane space. ATP synthase here in orange, or complex 5, and again this is a multi-protein complex, not an individual protein. It can allow a proton to move back from the intermembrane space into the matrix, and when it does that, it will synthesize a molecule of ATP, okay, from ADP and inorganic phosphate. So the substrates for this are ADP and also an inorganic phosphate. Okay, right, so that is my overview then of respiration. So, what is the effect of calcium coming into the matrix of the mitochondria on this respiratory reaction? Okay, well, it's going to speed it up, but which specific portions of this reaction are going to be sped up? Well, it's the Krebs cycle and also the pyruvate dehydrogenase. These are the portions that are going to be sped up, not the electron transport chain. Okay, but of course, downstream of these being sped up, you'll speed up the electron transport chain. So basically, pyruvate dehydrogenase gets activated by increased calcium in the matrix of the mitochondria. Okay, in addition, the Krebs cycle down here gets activated by increased calcium in the mitochondria. Okay, so the link reaction is going to be sped up. You're going to be taking more pyruvate, making more acetyl-CoA. The Krebs cycle is going to be sped up. Okay, you're going to be making more reduced NAD and more reduced FAD, and therefore the electron transport chain will be sped up because you've now got more NAD and more reduced FAD. Okay, and that means that you're now going to have more protons being moved from the matrix to the intermembrane space, and therefore ATP synthase activity will go up and you'll create more ATP. Okay, so for those of you who want to know the specific enzymes of the Krebs cycle that are going to be activated by calcium, they are alpha-ketoglutarate dehydrogenase. Okay, so if you're familiar with the enzymes of the Krebs cycle, then you might be interested to know that it's alpha-ketoglutarate dehydrogenase, which I'll just abbreviate down to DH, and also isocitrate dehydrogenase, which are actually activated by increased calcium levels within the matrix of the mitochondrion. Okay, for those of you who aren't familiar with the enzymes of the Krebs cycle, then just know that calcium is going to speed up several important enzymes within the Krebs cycle, and therefore is going to increase the rate at which you're forming reduced NAD and reduced FAD, and therefore is going to lead to increased electron transport chain and increased ATP synthesis. Okay, right, so that now concludes our discussion of how calcium going up in the cytoplasm is going to lead to calcium going up in the matrix of mitochondria, okay, and what effect that's going to have on mitochondrial ATP production. In the next video, what I want to do is talk about a really key example of IP3 receptor mediated calcium signals, okay, which is in the contraction of smooth muscle cells. Okay, so we'll do that in the next video.